Seed dormancy and germination. In plant kingdom, seed-bearing plants, also called as spermatophytes, they produce seeds after pollination and fertilization. Spermatophytes include angiosperms, which produce enclosed seeds, while gymnosperms, they also produce seeds but are naked. The seed, one can define seed as fertilized ovule. Seed has testa, seed coat, endosperm for storing food, and dormant embryo. Seeds, after getting favorable conditions for germination like light, temperature, oxygen, water, germinate and produce seedling, which has a radicle, that is root, and plumule, that is stem, and cotyledon. In dicotyledonous seed, two cotyledons, and in monocotyledonous seed bears single cotyledon. Germination of seed is controlled by seed dormancy. Seed dormancy actually is a state of seed in which it is prevented from germination even under favorable environmental conditions. In brief, one can define seed dormancy as the tendency of seed to prevent its germination even under suitable conditions. During seed dormancy, all metabolic activities are in the stationary phase. Seeds adjust their dormancy status by sensing a range of environmental signals. These signals trigger switch on and switch off mechanism of dormancy in seeds. Some plant species have seed dormancy of few days or few months, while the dormancy period of about up to 50 years is also there to some of the seeds. The seeds of fruit plants, for example mango or citrus, they germinate immediately after extraction from the fruits. While in case of apple or pear, the seeds do not germinate for several years. Dormancy in seeds is an environmental adaptation of the plant to survive under stress conditions. So, one can define the dormancy as a state of metabolic arrest that facilitates the survival of seed during unfavorable conditions. Seed dormancy is affected by endogenous and exogenous factors. like hormones and environment. Now, what is the importance of seed dormancy? That seed dormancy is very important in the life of every plant because the plants can overcome the unfavorable conditions and perpetuate in future, and this thing will happen only if there is dormancy phenomenon. Also, dormancy plays important role in delaying germination, which allows dispersal of seeds and at the same time prevents simultaneous germination of all seeds in nature. It also helps to avoid self competition among the plants. The longevity of seed dormancy differs in plants, or the duration of seed dormancy is different for different plants. Some of the examples we will see. In case of oxalis, there is no seed dormancy, while in salix, dormancy period is of about 12 hours only. A plant like poplar, the seeds have dormancy of a week. Then in case of pine and spruce plant, dormancy of 4 to 5 years. In case of xanthium, this dormancy goes up to 20 years. In case of Indian lotus, dormancy recorded is up to 1000 years. So this is related to the duration of seed dormancy in different plants. Another importance, due to dormancy, seeds gets prevented from germination during unsuitable ecological conditions like drought, high temperature, etc., when there is very low survival of the plants. So in such situation, due to dormancy, the seeds remain alive. Due to dormancy, there is staggering seed germination, which is safeguard the species from damage or death in harsh ecological conditions. Dormancy also helps seed to germinate when there is less competition of resources like water, sunlight. Storage of food grains is possible only because of seed dormancy. As a result of this, we are able to store our food grains, pulses, or oil seeds for specific period. In agriculture, seed dormancy plays crucial role. Because of this character, we can sow the seeds in next growing season to raise the new crop. Seed dormancy is the survival mechanism in the plants. So these are the importances of seed dormancy. Now, types of seed dormancy. So, exogenous or also called as ectogenous dormancy and endogenous dormancy. So, exogenous seed dormancy is imposed by external factors outside the embryo. The tissues enclosing the embryo affect germination by inhibiting water uptake, providing mechanical resistance to avoid the embryo expansion and radical emergence. It also modifies gaseous exchange. It prevents the leaching of inhibitors from the embryo and supplies inhibitors to the embryo so that embryo does not germinate. This type of dormancy, that is exogenous dormancy, is subdivided into three types as physical dormancy, also called as seed coat dormancy, mechanical dormancy and chemical dormancy. So, physical dormancy or seed coat dormancy. Seed coat, that is covering of the seed, becomes hard, fibrous or mucilaginous during dehydration and ripening. As a result of this, seed coat become impermeable to water and also to gases which inhibit germination. This type of dormancy is very common in case of drupe type of fruits, for example olive, peach, apricot, plum, etc. Seed coat becomes hard and suberized in many legumes, preventing seed germination. As the hard and thick seed coat becomes impervious to water and gases. It is mainly due to water impermeable layers of palisade cells in the seed coat or fruit coat. Such seeds prevent uptake of water and avoid seed germination. The Malpighian cells existing in many layers of palisade cells found in exotesta forms a impermeable layer for the entry of water in seeds and hence seed remain dormant without germination. Physical dormancy is also imposed by chalazal plugs in seeds. For example, Bixaceae and Malvaceae members. In these plants, such chalazal plugs do not allow water to enter in the seeds and force the seed to remain dormant. After physical dormancy or seed coat dormancy, another exogenous dormancy type is mechanical dormancy. Here, due to very hard seed coat, seeds will not germinate unless and until seed coat is softened, either by creating moist and warm conditions in storage or by microbial activity. The seed covering restricts emergence of radicle and thus inducing seed dormancy. For example, walnut. So seeds of this plant are having shells and stones which are too strong to allow embryo to expand during germination. This type of dormancy is due to mechanical resistance of seed coats, shells or stones of different fruits. Third type of exogenous dormancy is chemical dormancy. So various chemical inhibitors like phenols, abscisic acid or coumarins, they are responsible for chemical dormancy. These germination inhibitor compounds are present in seed covering tissues and inhibit seed germination, keeping them in dormant conditions. Many fleshy fruits in which their seeds remain in juice, for example citrus, cucurbits, grapes or tomatoes, etc., exhibit chemical seed dormancy. After taking survey of exogenous dormancy, now endogenous seed dormancy. So this type of dormancy is imposed by rudimentary or undeveloped embryo at the time of ripening or maturity. Embryos are not completely developed, and due to this, such endogenous type of seed dormancies are observed. So it has different types like physiological dormancy, morphophysiological, combinational, that is combination of exo and endogenous seed dormancy, and secondary dormancy. So these are the types of endogenous seed dormancy. At first we will discuss physiological dormancy, a type of endogenous seed dormancy. Physiological dormancy is most prevalent and very common type of seed dormancy and is widely seen in different plant groups of gymnosperms and major families of angiosperms even. Some of the examples are Lactica sativa, sunflower or tomato plant Lycopersicum esculentum, and also in Arabidopsis, and many cereals they also show physiological dormancy. This physiological dormancy is also classified into different subtypes like deep, intermediate, non-deep, photodormancy, thermodormancy, epicotyl dormancy, double dormancy and secondary dormancy. So these are different subtypes of physiological dormancy. And physiological dormancy is one of the type of endogenous dormancy. So one by one, what are the types of physiological dormancy that we will study in brief. First is deep dormancy, So here seeds have deep physiological dormancy, and this will be when if embryos excised from such seeds either don't grow or will not produce normal seedlings. Instead, seedling develop will be abnormal. GA treatment, if one give to the seed or embryo, then even this dormancy is not breaked. 3 to 4 months of cold or warm stratifications are required before germination can take place. One of the example of deep dormancy is acer platanoids. So 3 to 4 months of cold or warm stratifications are required to break the deep dormancy. So stratification means placing of seeds close together in layers in moist sand or pit to help them germinate. Now, subtype of physiological dormancy, that is intermediate dormancy. So seeds having intermediate physiological dormancy produce normal seedlings when embryos are excised from such seeds. If one give gibberellic acid treatment, it promotes germination in most of the seeds, but not all species. Such seeds having intermediate dormancy require 2 to 3 months of cold stratification. Dry storage can shorten the cold stratification period. And example, acer pseudoplatanus. Third type, non-deep dormancy. So seeds with non-deep dormancy produce normal seedlings when embryos are excised from the seeds. Gibberellic acid treatment here can break the dormancy. Depending on the species, dormancy can be broken by stratification. Fourth type is photodormancy. So, in such type of seeds, they require light or dark condition for germination. Without these, they are unable to germinate. Such seeds are known as photodormant. Dormancy is mainly controlled by photochemically reactive pigment known as phytochrome. Such phytochromes are widely present in plants. When imbibed seeds are exposed to red light having wavelength range in between 660 to 760 nanometer, the phytochrome changes to red form, also called as a PR form, and thereby substitute the process of germination. Fifth type, that is thermodormancy. Such seeds germination is dependent on specific temperature. Without that, they do not germinate and remain dormant. This dormancy is known as thermodormancy. Example, lettuce seeds. Epicotyle dormancy. Special type of dormancy in which seed germinate but epicotyle remain dormant in absence of chilling treatment. For example, polygonum. Double or combinational dormancy. So this dormancy is due to combination of ecto and endodormancy, hence known as combinational or double dormancy. Some tree legume species, they show this type of dormancy and have hard seed coat and dormant embryo. So such plant seeds require 2 years for breaking the seed dormancy. In first year, some microorganisms acts on impervious hard seed coat and then make it soft and pervious to water. In next year, due to chilling treatment in the winter season, the embryo dormancy is removed and then seed germinate. Such seeds have such double dormancy. To break the dormancy, these seeds require combination of two different treatments. Some seeds require stratification followed by exposure to light or cold treatment. Now, imposed dormancy, another type. Seeds are imposed by different environmental factors like temperature, water, light. Dormancy due to metabolic blocks. Such type of dormancy is seen in some seeds where metabolism leading to germination of seeds is blocked, and hence this type of dormancy is known as a dormancy induced by metabolic blocks. Then innate dormancy, also known as a spontaneous dormancy. It is due to physical, physiological, morphological, mechanical and chemical factors. And now, secondary dormancy. This dormancy is due to unfavorable conditions of germination in imbibed seeds, which have absorbed water and ready to germinate, but it avoid to germinate if there is very high or low temperature outside, water stress, and also prolonged darkness. Many factors are involved in seed dormancy. Overall, we can see the factors like temperature or chemical, light, genetical factors, physiological factors, molecular factors, also anatomical, morphological, embryological, ecological, also we will call it as environmental factors. They are involved in seed dormancy. Now we will go towards how this dormancy period can be broken. Actually, this is a gift to the plant seeds given by the nature, that is the dormancy. To have dormancy, what is the significance of dormancy that we have seen? So under unfavorable environmental conditions, the plant can survive, and under favorable condition, it can germinate, and this is a very nice event or the phenomenon we can say, that is the seed dormancy. But even though if one is in need to produce plant from such dormant seeds, what to do for this thing? So researchers, they have tried, and they have given some of the methods to break the seed dormancy. So here one can break the seed dormancy by stimulating the embryo metabolism, by increasing permeability of the seed coat to oxygen and water. Also, one can reduce the mechanical resistance to enhance the growth of embryo. One can reduce the influence of inhibitors of germination like hormone abscisic acid, or one can use a hormone or synthesize the hormone to initiate the germination. So like these efforts are being made by the scientists. And in such case, efficiency of treatment or its degree, that depends on kind of dormancy present. So in some seeds, this dormancy of seed can be broken by any one of the available method. For some kind of dormant seeds, only specific methods are required. While in some dormant seeds, it cannot be broken by any method or common treatment at present we are using. So we will take survey of common methods of breaking seed dormancy. So first one, that is stratification. Second is scarification. Then by giving water treatment. Fourth is by light treatment. So one by one we will go towards what are the methods of breaking seed dormancy. So first, stratification. So embryo dormancy is commonly broken by stratification method. By storing the seeds at low temperature, usually 1 degree to 5 degrees Celsius temperature is for period of about 30 to 120 days. While chilling up embryo for 60 days, in case of apples, remove the seed dormancy. So this method of stratification is very commonly used to break the seed dormancy in forestry and horticultural plants. So stratification we have seen means placing the seeds in layers of moisture retaining media such as peat, moss, sawdust or sand along with low temperature and moisture. When seeds are stratified, they are kept in plastic bags and stored at low temperature. These plastic bags they are opened periodically for aeration. So second method of breaking seed dormancy after stratification we will study is scarification. So some of the seeds does not contain any germination inhibitor, but its seed coat is very hard and impermeable to water. For example, members of legume family plants, seeds. In their natural environment, these seeds are waiting for drenching spring rain that softens hard seed coat and allow the water to enter the seed, which stimulate germination. Many times these seeds are scarified by nicking the seed coat with sharp knife or razor or by using sandpaper scrubbing. Scarification is the process of breaking, scratching, mechanically softening the seed covering so that it becomes permeable to water. There are types of treatment used for scarification, like mechanical scarification, scarification by using chemicals, or scarification by use of hot water treatment. So first subtype of scarification we can say mechanical scarification. One of the simple and effective method of scarification. Hard seed coat is chopped by rubbing with the sandpaper, cutting with file or cracking with hammer. It is used for small amount of seeds. And the seeds having large size. For large scale scarification, seeds are tumbled in drum lined with sand, paper, or in concrete mixers containing coarse sand or gravel. Stratification should be done carefully so that seeds are not injured or damaged. and the inner parts of seed should not be exposed. Another type of scarification is acid scarification. So by using some chemicals, dry seed coats are softened. For example, use of concentrated is sulfuric acid or hydrochloric acid. So in this method, one part of seed and 2 parts of acids, this ratio is used. So at a time 10 kg of seeds are treated. The containers of glass or wood or non-metal or plastic are used. Mixture is stirred continuously during treatment to have uniform results. Time of treatment depends on seed type, and usually it is 10 minutes to 6 hours. So after treatment, acid is removed and seeds are washed carefully till the acid is removed completely. Treated seeds can be sown immediately or can be dried and stored for future use. For example, seeds of legume, also seed of bajra, brinjal and tomato are given treatment of sulfuric acid and good responses or results are seen. Another method of scarification, that is hot water scarification. Seeds are given hot water treatment in the range of temperature in between 77 degrees Celsius to 100 degrees Celsius. Seeds are soaked gradually in hot water for 12 to 24 hours. Solan seeds are separated and are immediately sown after hot water treatment. Warm moist scarification now. In this method, seeds are placed in moist warm medium for many months to soften the seed coat and seed covering through microbial activity. Method is beneficial in seeds having double seed dormancy. So up till we have seen methods to break seed dormancy. First one, stratification. Second, scarification. Now, use of water. By using water, how seed dormancy can be broken, that we will see. So soaking of seeds overnight in water can soften the hard seed coat enough to allow moisture inside the seed so that seed can germinate. Or also one can use boiling water instead of normal water. So by this method, different type of legume seeds dormancy can be broken by using light. Now another method. So here germination inhibitors in the seeds especially in case of wildflowers. This dormancy can be broken by exposure to the light only. Then double dormancy. Many seeds require combination of treatments to break their dormancy. These types of seed require stratification as well as followed by exposure to light or sometime treatment with moist soil followed by cold temperature. By using such combinational methods, double dormancy can be broken. Dry storage is the another method. It promotes ripening in certain seeds which are dormant and freshly harvested. Freshly harvested seeds of many annuals and herbaceous plants fail to germinate until period of dry storage. Now treatment with different chemicals. Some of the chemicals like potassium nitrate or gibberellic acid are given prior to sowing to such seeds and are able to stimulate germination of different seeds. For example, sweet orange or sour orange or citrus species seeds are given gibberellic acid treatment for their germination. Another method of breaking seed dormancy is treatment with growth regulators. So use of particular low concentration of growth regulator like gibberellic acid or cytokinine, ethylene, etc. can break the seed dormancy. Chilling treatment is another method of breaking seed dormancy. So in many plant species, seed dormancy can be broken by giving chilling treatment, and where seeds are stored under temperature range from 2 degrees Celsius to 5 degrees Celsius. After treatment, seeds are ready to germinate. Chilling treatment time and temperature range varies from species to species.